Good. We'll have a quiz tomorrow. The quiz will start with last week's video and all of our work on prokaryotes and eukaryotes that we have so far. And it will include the resource that you made yesterday, all that information about what organelles are part of which kind of cell, and the cladogram that we looked at with the chart that we made to go along with that. So I would make sure I could do those things. And it will also include what we're going to look at today, which is how prokaryotes probably came to be, current theory on that. So this will be important as well. And I did give you a handout. You might want to make some notes as well. And they won't all be exactly what's on your handout, but that's a picture, so you won't have to draw this down. So you are aware of 3 types of cells. These should look familiar to you by now. Which one of these is the prokaryote? Which one is the prokaryote? Which one's the prokaryote? What? Why? So plant cells are prokaryotes?
Yes, also the cell wall.
Okay. So what's the way that you will tell a prokaryote from a eukaryote? What will you be looking for? So the vacuole is the big central vacuole. You'll see that in a plant cell. You won't see it in other things, though. So the cell wall is a characteristic of a plant cell, so is a vacuole. How will you tell the eukaryotes from the prokaryotes? We need to consult our resources from yesterday, because if you really don't know, this isn't going to go well tomorrow. We're talking about this last Friday. It's the big idea that you will look for. You might want to write this down in red somewhere. We're going to look for Ben finding as well. When we labeled the parts of these cells, we colored something. I colored it orange over and over and over again. Okay, we have 2 guesses. 3. Anything else? Really? Okay, 3 people have indicated that they might have 4. 5. Anything else? Rachel, you can look for. Okay, some more. You're on to something here. Okay, phone friend. All right, Ben, can you expound on that?
I was just trying to say that it is a membrane bound organelle and prokaryote.
Exactly. You're looking for that membrane and you're looking for it everywhere. Okay. So you might want to jot that down. Eukaryotes have membrane-bound organelles. It's kind of an important thing. Let's see if we can find them. In this plant cell, we have a membrane around the whole thing, which we also have in the prokaryote. But in the prokaryote, that's the only place we have a membrane around the whole cell. In the plant cell, we have a membrane around the cell. We have a membrane around the nucleus, the mitochondria, the chloroplast, the Golgi apparatus, all the endoplasmic reticulum, the nucleus. Same in the animal cell, except there's no chloroplast. Membrane around the whole thing, membrane around the nucleus, endoplasmic reticulum is all membrane. We have vesicles. There is a membrane around all of the mitochondria. Much membrane here. So membrane-bound organelles. All of the parts have membrane around them. Think of it like this. Okay, you're going to bring your lunch to school, and you're a prokaryote. You're going to get a bag, and you're going to throw in a couple of slices of bread. Okay, maybe a couple of slices of lunch meat. Maybe a big glob of mustard or ketchup, some cheese. And you're going to maybe dump in some potato chips on top of all that. And then, I don't know, maybe you'll throw in some grapes or a banana, maybe some carrot sticks. Okay, and then you're going to drop a cookie on top of all that in the bag. Get it? You get this bag full of stuff. Now, if you're a eukaryote and you're going to make your lunch, you're going to get out the pieces of bread and you're going to put the lunch meat on them and the cheese and the ketchup and mustard and mayonnaise or whatever you put on sandwiches. And you're going to put them in a baggie, and then you're going to put that baggie into the bag. See where we're going with this? Okay. Then potato chips, you'll do the same thing. You'll get a couple of handfuls of potato chips out of the bag. You'll put them in a bag, and you'll throw those in. Carrot sticks, the same thing. You'll put those in a separate baggie. So instead of having one bag with all this junk in there, all jumbled around, like we have in our prokaryote, you're going to have neatly packaged stuff in the eukaryote with a plastic bag membrane around them. So that's kind of huge that you write down. Eukaryotes have membrane-bound organelles. So you put that somewhere and make sure it's a big deal. Put it on your stuff from yesterday. So why then do some cells have everything neatly packaged as eukaryotes, and why are some prokaryotes? Okay. Here's your big definition of prokaryotes and eukaryotes that you need to make sure you know. And the important part is eukaryotes have membrane-bound organelles. This is the big deal. Prokaryotes lack those things. Students tend to just identify the nucleus as a membrane-bound organelle, but there are many others, and we do not necessarily have the nucleus. The fact that there's a membrane wrapped around everything. Think about the lunch bag. Okay. Eukaryote cells have membrane-bound organelles. Prokaryotic cells do not. They just have a membrane around the whole cell, like your lunch bag. Okay. So how do we get eukaryotes? This part you might want to jot down. I'll give you a moment to look at what all this means. This probably should go in your notes, the origin of eukaryotes. A lot of big words here. We'll stop and talk about those. Ancestral chloroplasts were photosynthetic. Okay, were photosynthetic prokaryotes that became endosymbionts. What does that mean? It means that the chloroplasts were individual little prokaryotic cells. They didn't have a nucleus. And they suffered a fate of becoming an endosymbiont, which we'll see what that is. They were ingested by another parasitic cell. They were ingested. Somebody came up and engulfed them and did not digest them. They stayed in the cell. Why weren't they digested? I don't know. Maybe their membrane was just resistant to anything that the ingesting cell could produce. So what happened? Here's this in pictures. Is we had a cell, and for some reason or another, maybe because it ran into things, who knows, it began to get some little dents or depressions or infolding. This is membrane infolding. And that membrane then, as it infolded, allowed other things to come into the cell. So membrane infolding is what formed as the membrane unfolded. It first encompassed the nucleus, and then it may have formed all of that endoplasm in particular and all of those leaky things outside the nucleus. So looking at all those big words, we'll stop and make sure you know what they mean after you jot them down. So this is kind of the big idea here. Let's look at these words. Ancestral, you know what that means? Okay, what about a chloroplast? Where do we find those? What kind of cell? Okay, and what kind of eukaryotic cell? Do animals have them? No, plants do. Okay, so these chloroplasts are just floating around in the world doing photosynthesis, and a prokaryote came along and engulfed them. That's the gist of this, okay? We're going to look at that in detail. Good? Membrane infolding, just as dense in the membrane that things can go in. And as something would creep into the membrane, it could engulf something that's already in the cell, which is what happened to the nucleus. There's DNA floating around, the membrane came in and eventually wrapped itself around the nucleus because it was an advantage. Now, everything we're looking at here, all right, and this is huge. You may already have some ideas about what is a hypothesis, a law, and a theory. Let's hear this first. Hypothesis, what is it? What's hypothesis? What's hypothesis statement? Okay, what else we got?
Theory.
Okay. What else? Do you have no idea what a hypothesis is? You've seen this before. Come on. Question. And what about theory? What do we got for theory? What theory, what do you think of? What do you think of when you think of theory? What do you think of? Jasper. Okay, what else do you think of? What else do you got? Okay. And what about law? What about law? What do you think of when you think of that? Okay. So after you finish erasing all that stuff from your heads, I'll tell you what they are. Okay. And you're going to spend some time looking at them on Friday as well. So a hypothesis is a testable statement of what you expect to find, maybe. And it must be testable. You must be able to say yes or no, this happened. You must be able to say that when you're done carrying out your trials. You must be able to say, yes, my hypothesis was supported. The evidence shows that that's what happened, or, oh, no, it didn't. Either is okay because now you know what didn't happen. You're going to be able to say, well, yes, indeed, that's what happened. That's how it worked. Or, no, that's not all what happened. A law is something that you observe repeatedly many times, happens the same way. It's often an equation. Example of that, and I'll use a physics book to show you that example. What's going to happen if I drop this physics book? How do you know? Oh, I'm glad you said that. Thank you. So there is a law of gravity that says if you're on Earth and you're relatively close to Earth, you're not in outer space or something, and you drop something, it's going to fall to the ground. Okay. Theory of gravitational attraction will explain why it does. Theory of gravitational attraction says because the two masses have an attraction for one another. The law says what happens because we've observed it so many times. A theory explains why it happens. So the theory takes many, many, many observations and many, many, many things that we know. Okay. There's nothing unknown about a theory. We say, well, because of this, this, and this, things fall to the ground. We usually observe the law first. That's usually the first thing that happens. The next thing that happens is after we know a whole bunch of stuff, we test it. And then lastly, we explain it. So we observe, we test, and then we explain. How's that working for you? Okay with that? Okay, so why we're doing this is because the idea of endosymbiosis and how eukaryotes came to be are a theory. They explain a bunch of things that we observed. So as we work through this, I want you to look for the evidence. Look for the evidence. Okay, I want you to look for the evidence as we work through this. Okay. So here's your handouts. All right. The person that initially made this, who isn't me, thought this is a hypothesis. They're wrong. It's a theory. So this is your handout, and this is what's going on in your handout. We're going to take a look. We have a couple of things going on here. We have an aerobic bacteria, and please write all over this and do whatever you need to understand things. So you might want to note that aerobic means it uses O2. like when you do aerobic exercise, you're taking in a lot of oxygen. Aerobic uses O2. And so the aerobic bacteria gets ingested by a prokaryote. Okay? It's ingested. That happens by infolding, just like the nucleus happened. The aerobic bacteria is ingested by the prokaryote. Okay? Now, one of a couple of things can happen here. In part B, we see that this aerobic bacteria has now become a mitochondria. The aerobic bacteria is now mitochondria, which makes sense because you probably know that the mitochondria takes the glucose, uses oxygen, and gives you energy. ATP is involved, a lot of ATP. Okay with that? Right. Another point then, the new cell, the prokaryote that has ingested some aerobic bacteria, is now an animal cell. And many different things can happen to that animal cell at this point. If that bacteria, then that initial prokaryote ingests something called a cyanobacteria. Okay. Cyanobacteria is blue-green algae. If it gets ingested, the blue-green algae can do photosynthesis. This is probably the first chloroplast. Following so far, or questions? Okay. So again, we're going to look at some evidence here. We have our hypothesis, our law, and our theory. Okay. So we observed repeatedly over and over again that there's something different about a chloroplast, a nucleus, and a mitochondria. We then tested that by looking at all of the chloroplasts and mitochondria and nuclei in the plant and animal world. Fine. So we had a lot of tests, and we found, yes, indeed, they all do have two membranes. And then we said, well, here's how that probably happened. Now, this theory is pretty good. There's not another one that explains the existence of those double membranes or even the existence of eukaryotes so well. So we live with that. And here's our evidence. Aerobic bacteria probably became mitochondria, cyanobacteria became chloroplasts. Now, the other interesting thing here that's kind of seals the deal is that mitochondria have a double membrane. They have DNA as well. They have their own DNA that is different from the nucleus of the cell. So they have different DNA. How do they get that DNA? They had to come from somewhere else. They have different DNA, and they have a double membrane. We observe those things repeatedly. We don't know currently of a mitochondria that doesn't have two membranes and different DNA from its cell. We don't know of a chloroplast that doesn't have double membranes and different DNA from its host cell. Are we okay so far? I'm kind of concerned because we couldn't even remember the difference between a prokaryote and a eukaryote. Now I'm asking you a bunch of stuff. All right, I'd like you to turn to somebody nearby, one person or maybe two. And I would like you to explain the evidence that we have for endosymbiosis. Explain the evidence that we have for endosymbiosis. Find a person, stay distant, but talk. Just for the evidence. Evidence. What did we first see that made us think that this might happen? That's true. But what you're saying this way was it. First made us go, hey, you know what? I'm looking for this.
What's the evidence for
What's our evidence?
The prokaryote broke down, and then bacteria and eukaryote.
What's our evidence? What made us go, this is probably happening?
are the steps?
The role that is introduced and the prokaryote, so maybe the problem happens is it's like
We see. For evidence.
The aerobic bacteria. Oh my God. Okay. Since the aerobic bacteria got broken down, they became mitochondria, and once the mitochondria got broken down,
Okay, let's have a little evidence discussion here, okay? So somebody's wondering, how did eukaryotes come about? And they're looking at eukaryotic cells and they're looking at prokaryotic cells. What did they see about the eukaryotic cell that was interesting? What did they see? Yeah, we saw some organelles, namely the chloroplast and the mitochondria and the nucleus, all had double membranes. Okay, double membranes. Also, upon analysis of mitochondria, chloroplast, and nucleus, we found that they had different DNA. Chlorophyll had one pattern of DNA, the mitochondria had another, and the nucleus, which was what the cell replicates, had still another. Okay. So we looked at that and went, how could that have happened? These three main organelles all have extra membrane and they have DNA that's different from one another. How did that happen? So that's our evidence. That's what we know. What we then said is, well, it could have happened like this. It could have happened like this. So our theory, our explanation is the endosymbiosis part. The observations were, when we looked at cells, we saw those three organelles. What were they again? Double membranes, three organelles, what are they? Mitochondrial, chloroplast, and the nucleus. All have double membranes around them. And then, upon looking further at the cell, we could see that this cell has a lot of membrane in a lot of other places too. Look at all the membrane here that's really just endoplasmic reticulum, Golgi complex, rough and smooth endoplasmic reticulum. All the vesicles and vacuoles all have membrane around them. So we were then able to deduce that by infolding, which is where there's a dent in the membrane and something is kind of engulfed, by infolding, we created a bunch of membrane in the cell, and we were able to engulf a chloroplast if we were a plant, and a mitochondria if we were an animal, a chloroplast and a mitochondria if we were a plant. If no chloroplast was engulfed, then we had an animal cell. So are you clear on what's the evidence and what's the explanation? Okay, take a moment and write that in your notes. Evidence for endosymbiosis, and then explain how that happened. Take a moment and summarize that for yourself, and then we'll share those. Evidence and explanation. Evidence, what's the explanation? How did this? Make sure you can cite the evidence. You're looking for double membranes. We really couldn't verify the different DNA until about the 1950s, maybe, when we were able to do some analysis of the sequence of all of the nucleotides. So how about you prepare for this quiz tomorrow? And I also study getting that. Study isn't specific. So one professor that I had in college in my first master's degree said, to study for stuff, if you're going to have a test, try to second guess what kind of questions you might be asked. So if I were the teacher, what questions would I ask about this, and then make sure you can answer them. So think about, for this, think about what's the name of the process, how did it happen? What's the evidence we had? What made us look for this? And why is it a good explanation? Difference in prokaryotes and eukaryotes. You're going to want to quiz yourself on the parts. The good way to do that is maybe to practice drawing each kind of cell, okay, and see what you put in there, and then check yourself. Make sure you can label those main parts that we looked at: nucleus, chloroplast, mitochondria, cell membrane, cell wall. Make sure you know the differences, what isn't in prokaryote, what is in the eukaryote. Just start with all your stuff. Maybe rewatch that video from last Friday, which is still there. Last Friday's video was to introduce you to this, but a lot of it might make a lot more sense now that you've heard a lot more. The second time around might be a good listen. Archaea, extreme environment. What kind of extreme environment? So we listened to that video maybe. It's not very long, was it 12 minutes or something? It's not very long. So listen to that again. Look at the work you did in answering those questions in that video on Friday. Look at our work from the last couple of days. Remember, if you want to do last Friday's quiz, you get a different version, the paper version, and you are completed. So I'm also reviewing my slides. Also, it didn't make work, and some of you have none, and that's awesome. If you do, the 9 weeks ends on November 6th. And there is no living way in hell I'm going to spend that last week before November 6th grading all the stuff that you thought I was doing in the meantime. So if you come in to me by this Friday, I will evaluate it. If not, it comes in late next week. I might do something with it, I might not. So no guarantees. So for that test tomorrow, you have a video and some reading for Friday, and then you have something you need to do, okay? Bye, have a good day. Good morning. How are you? Maybe I'm not sure. I'm not sure. So we'll see if we get through this today. We'll do the other set of mastery questions tomorrow, and you have a new adventure on Friday. See where we get with this. You guys are smart. Hello.
I thought you were going to call it slow at first, not smart.
How did you even think that? No, no. very methodical in the way that you think. You work right through stuff. It's all good. So a couple of things on this handout. You might want to take the last page off so you can refer to it more easily. It's just kind of your guideline for this whole thing. And let's make sure that you understand these terms. Then let's look at how you'll want to do this. So these are some possible things that when we've looked at all of them at one point or another, that you will want to use while you describe and explain what happened. And you're going to want to also probably have your copy of those 5 of those principles. Like utilitarianism, that you want to have that list as well so that you can have them handy. You won't have to try to remember them. You can look at those to jog your memory. It's about memorizing this stuff. It's about being able to use your references, your resources. We don't have a lot of quizzes. If you want.
Yeah.
About being able to use a resource rather than memorizing junk. So let's make sure we know all of these terms. If you look at one of these terms, can you have something in your head about what does it look like? Like solidification. And you might picture magma coming out of the earth, and then when it gets to the surface, it's lava, and then when it becomes solid, it's like basalt or obsidian. It's a rock. So that's what you would see with solidification. What about deposit? If something is deposited, what are you picturing? Okay, good one, good one. What's unconformity? What do you picture there?
Like yesterday, the picture showed all of the layers curved, and then the top one was straight.
Okay, and stop the recording. So that's something.